We're going to be talking about enzymes today. Okay, the query came up of the whole sort of reaction thing is a bit confusing. And what I find is that people often get confused between mode of action and course of reaction. So in this lesson, we're going to go through both of those to hopefully point out the differences and get everything straight in your head. So just to recap, when we learn about enzymes, we're going to be learning about mode of action, course of reaction, and factors affecting reactions. So this one is when we're talking about temperature, pH, substrate concentration, and enzyme concentration. And that's what we've actually focused our enzyme live lessons on up till now. So today we're going back to the basics and we're going to look at the mode of action and the course of reaction. So if we look at it most briefly and sort of at a large overview, mode of action, this is how an enzyme gets the job done. So one particular enzyme, how does it go about catalyzing one particular substrate? Course of reaction is slightly different. When we're talking about a course of reaction, we're talking about many enzymes. We're talking about an enzyme-controlled reaction in general. So this is how the enzyme reaction progresses. So there are overlap a little bit, but there's also some really important differences. And it's important to get your head around them, because when it comes to answering questions, you'll get questions on mode of action and you'll get questions on course of reaction, and you need to be able to answer specifically which one it is that's being asked. So before we get into the details of these, I just want to touch base and make sure that we are absolutely crystal clear on the basics of what is an enzyme. So there's a few key features here that are critically important. Your enzyme is a biological catalyst. Okay, how does it do its job in general? Well, what's the point of an enzyme? Remember that in chemistry, reactions happen, okay? But when you're looking at living organisms, reactions have to happen at a speed which can sustain life. And that's where we have this problem of activation energy. We need energy to make a reaction happen. So in chemistry, maybe we put things in a boiling tube and hold them over heat. Well, we can't exactly boil our finger every time we want our muscles to move, for example. So we need some way of making these reactions happen in a time frame and in conditions that are acceptable for living organisms, and that's where enzymes come in. They lower that activation energy that's required to make the reaction happen. So they reduce activation energy to increase speed of reaction. They are unchanged by all of this. Remember, a catalyst just does its job, carries on and does the same job again. The enzyme itself is not involved in the reaction, it catalyzes the reaction. Okay, if we take it back a step, enzymes are globular proteins. They have a very precise 3D shape. So we are talking about tertiary. Okay, that's the further folding and coiling of our protein. After it's been through primary and secondary, then we're coiling and folding it further to make that blobby globular protein. Okay, in some cases, we also have quaternary structure. So our enzyme could have multiple polypeptides, it could have prosthetic groups, but it always will have tertiary. So all enzymes have to have tertiary in order to get that globular shape. Some enzymes have quaternary on top. What is special about this specific precise shape, in particular for an enzyme, right, is not just a globular protein, not just a precise shape, it's this active site. Okay, what is the active site? The active site is a cleft or a depression. So if we've got our globular protein, the active site dents in. So things can fit into the active site. Okay, the active site has an extremely precise shape. You wouldn't say extremely in your exam, you would be nice and clear and technical, precise 3D shape. Okay, and this shape is specific, okay? It's not only precise, it is specific. And it is specific to a particular substrate. Specific shape, complementary to the substrate. Now, as we go through mode of action and course of reaction, you're going to see things of this list coming up. So it's important that you sort of get them set in your mind. This is an overall description of enzymes in general. Okay? We're not talking specifically about mode, we're not talking specifically about course of reaction, we're not talking about factors. This is just an understanding of enzymes. So yes, they can be overlap with other things, but have them split as well in your head. So there's your enzymes, really specific. Globular proteins, active site, precise shape, they're biological catalysts, so they reduce activation energy, they're unchanged by this whole process, they're not involved in the reaction, they don't take part, they don't react with anything, they catalyze a reaction. So, how does that actually happen? Mode of action of enzymes. So this is all about how the enzyme gets its job done. Okay? We are talking about one enzyme doing its job. So it's rather unrealistic in the terms of living things, but we need to understand how an enzyme works. So let's put a little note here. One enzyme doing its job. So different enzymes catalyze different reactions. Remember we said they are specific. They have a specific shape which is complementary to their substrate. So if it's an enzyme A for reaction A, it has nothing to do with reaction B. That needs enzyme B. So we can break bonds, we can form bonds, okay? We can have condensation reactions and we can have hydrolysis reactions. All of these are enzyme controlled, all with different enzymes for different reactions. So we can break substrate, we can join substrates, different enzymes for different reactions. But what are the specifics? Okay, so now we are talking about reaction A and enzyme A only, and we're looking at one little enzyme A. So we have said that the active site has a specific shape. That is part of the mode of action. That's a part of how the enzyme does its job. You wouldn't send a guy out to dig a hole without a spade. He needs that spade to dig the hole, okay? So the active site has a specific shape complementary to the substrate. So again, a note of being technical, being accurate. It's not the enzyme that's complementary to the substrate. It is the active site that is complementary to the substrate. Complementary means we can fit together. So when we fit together, it's not just a random in-out whether, I mean, it's just got to stay there, okay? So we have to have some kind of bond holding the enzyme to the substrate. So this is where we get our hydrogen bonds. So we should probably put in here, actually, substrate enters active site. Okay, so hydrogen bonds form between active site and substrate. So now our substrate is held in place, and that's where the magic happens. This is how we're going to lower the activation energy. So if we've got two things that we're joining, those two things can be held close to each other. So it's like making that reaction's like gonna happen. Okay, if it's something to be broken, that's one thing to start with, then what happens? We're gonna stress that bond, okay? And when we stress that bond, of course, we're increasing the likelihood that it's going to break. So we are lowering the activation energy of the reaction. So what we call this, we've got bonding, we've created something. This is what we call an enzyme substrate complex. Okay, and this is what happens to activation energy is lowered. Okay, when activation energy is lowered, the reaction can occur. So then products are created. So it might be a hydrolysis, we're creating two products. It might be a condensation, we're creating one product. Depends on the particular enzyme and the particular reaction that it catalyzes. Once the products are created, then the products are released. And remember that that enzyme is unchanged. So immediately those products are released, that enzyme is ready to catalyze the next reaction. So that one enzyme, enzyme A for reaction A, goes straight back to the top. We have a specific shape, complementary to the substrate. Substrate enters the active site and hydrogen bonds form between the active site and substrate, creating an enzyme substrate complex. Activation energy is lowered, products are created, then released, enzyme is unchanged. So the enzyme is like a little robot. He goes and he does the same thing over and over and over again. And this is the mode of action of enzymes. So there's a few things sort of to add around the side, which you could build in depending on your question. Okay, we talk about the enzyme being a specific, enzyme active site being specific shape and complementary to the substrate. So here, okay, we can also add in a little bit about our lock and key and our induced fit. So if the focus was on the active site and how these two fit together, we would bring in about our lock and key and our induced fit, because that's different ways in which we can understand and refine this specific shape and fitting together. When we talk about the hydrogen bonds forming between the active site and the substrate to form an enzyme substrate complex, if we have a more molecular twist on this situation, we would want to talk about the R groups of amino acids in the active site and parts of your substrate molecule. So it's not just random hydrogen bonds. Remember, all of this is specific. All of this is precise. All of this is very highly controlled. So it is specific R groups of specific amino acids within the active site, which are going to hydrogen bond to specific parts of your substrate molecule. Specific, specific, specific. That's the word for today. Okay, so then we can go through activation, energy lowered, products created, released, enzyme unchained, ready to catalyze the next reaction. So you've got that sort of basic core of the mode of action, and then you can add bits in to the sides, depending on what your focus is. And that, in a nutshell, is mode of action of enzymes. How one enzyme gets his job done. So the next sort of compare, contrast thing to look at here is the course of reaction. How is that different, and what is that in a nutshell, so that we can understand what the difference is with mode of action of enzymes. So the course of an enzyme reaction. What we're talking about here is many enzymes. Okay, so there's a few other things to bear in mind. So we're talking about many enzymes now, not just one. We're talking about a closed space. So a limited volume, for example. We're talking in terms of test tubes and beakers. We're not talking in terms of actual living cells, because there's a different set of parameters when you're in real life as to when you're in the lab. So when we're investigating things like this course of an enzyme reaction, we're talking about lab-based work. So because we have this closed space limited volume, we have a finite amount of substrate and enzyme. There is only so much substrate. There are only so many enzymes. So yeah, there might be thousands or millions of them, but there are only that many. Okay, what this means is that we can actually then go and look at, okay, so when we mix our enzyme and substrate together, what happens? Remember, here we're talking about lots of substrate, lots of enzymes. Mode of action was 1-1. Okay, it's all about probability. This is course of reaction is where we draw our 3 beakers. Three beautiful beakers. So we have an initial reaction. We have our middle phase, and we have our finishing phase. And there's different things that are happening in the different phases. So, in the initial reaction, we've got a high rate of reaction. In the middle phase, we have a slower rate of reaction. And in the finishing phase, we have a very slow rate of reaction. Okay, and why is that? So if we think about it at the very beginning, let's get some substrates. Okay, at the very beginning, we have 6 substrates, representing lots more than that. Okay, then we put in some enzymes. And you can see that the probability of an enzyme bumping into a substrate is very high. In the middle phase, we still have the same number of enzymes. In the finishing phase, we have the same number of enzymes. However, through this reaction, remember this is one test tube, we have less substrate. So the probability of bumping into a substrate molecule is decreasing, and that decreases the rate of reaction. So here we have lots of collisions. We have lots of enzyme substrate complexes. Now in your answers, you always write that out in full, and you don't use ES complexes. So this is one test tube. Okay, this is what happens in one test tube over time. The reaction only ends when there is no substrate left. So, mode of action, one enzyme, over and over and over again. That's happening on repeat within these course of reactions for each enzyme. So, the question then comes, and the question was actually asked today as well, is it the course of reaction that's affected by different types of enzyme or different factors? How do we measure the effect of different things on an enzyme reaction? We measure that on our initial rate of reaction. It all boils down to this one. Because this is the only place that we can see the changes. So what we're doing is, this is where we're looking at the effect of factors. Temperature, pH, okay, enzyme concentration, substrate concentration. Whenever you draw those classical graphs, you're looking at the initial rate of reaction and how that changes with pH or temperature. So we're looking just at this initial burst, this initial fast burst. Does it happen super fast, or does it happen fast, or does it happen not so fast because we've fried all our enzymes by boiling them? Okay, so the course of a reaction, yes, we study it and we understand it in full. But when we're looking at different factors affecting the reaction, then we're looking at the initial rate of reaction only. Okay, different types of enzymes would then be catalyzing different reactions. So each different type of enzyme would go through initial, middle, and finishing for its reaction. But they could have different reactions, they could have different speeds. Sometimes you have different enzymes catalyzing the same reaction, but they work at different speeds because they are different enzymes. Okay, they could also be affected differently by different factors. So each of them is going to have their own mode, which is going to be the same. We have a standard mode of reaction. Each of them is undergoing a course of reactions. But the effect of the factors, we're always looking in the same place, always at that initial rate of reaction. So that is everything for today. You've got your understanding of what is an enzyme. You've got your understanding then of what is a mode of action. How does this enzyme actually do its job? And then if we take it up a biggest picture to a course of reaction, then how we would tie back to the factors. So, if it doesn't make sense, then please make sure you do get it sorted out, because it's really important to understand this in order to be able to answer the questions.